United States Department of Agriculture.
Super, okay. So I will talk about three-dimensional fluid dynamic cancer model resembling the systemic drug administration for efficacy assays in the field of replacement of animal models. Basically, the approach we have used is to combine three-dimensional tissue models and organ-on-chip with the aim to overpass the limits of the current available experimental models, and in particular the use of animal models for such exploratory and drug testing assays. And in particular, we aim to have a fully humanized cancer model with the combination with the fluid dynamic system resembling the systemic drug administration for more predictive readouts. Therefore, the concept behind this research is the really combination of two important technologies of the last decades, which are the organ-on-chip on one side and the three-dimensional, in particular cell-laden hydrogels on the other side. And we focus the different cancer models, so I will not speak only about ovarian cancer model because this approach have been successfully validated in different tumor targets, which are the breast cancer, ovarian cancer, neuroblastoma pediatric cancer, and also pancreatic cancer. In all cases, our initial approach is to use cell lines, cancer cell lines, to validate the approach and to demonstrate the robustness of the approach. But it's also true that there are already other end users that are using the same approach using primary cells and patient-derived cells, highlighting the real strength of this approach. In particular, we typically use a cell-laden hydrogel based on alginate, which is an animal-free polymer structure, enabling it to grow and culture cells in a three-dimensional fashion more close to the in vivo condition. And compared to other polymers, this matrix has a very long-term stability, so we can also culture three-dimensional cancer models for many weeks and months with a tunable stiffness. This is really interesting. So you have the possibility to modulate the stiffness of the matrix according to the target of the tissue and supporting the proper cells proliferation and also migration and infiltration. It is also optically transparent and it is possible to collect the cells through the cross-linking of the matrix for further investigation. And this is also an interesting experimental point. Regarding the organ-on-chip, we have patented and developed a millifluidic device able to support the culture of these three-dimensional clinically relevant sites tissue models, because especially for cancer application, we do believe it's really important to keep the real heterogeneity of the tumor for such tests, because we know that also the drug response of cells might be really different if we consider the inner part of the cancer tissue compared to the external part, and also the chemoresistance and the drug resistance is really affected by the dimension of the tissue model itself. Therefore, this organ-on-chip that we have developed is not so over-miniaturized compared to other solutions to have the possibility to work with such three-dimensional tissue models and also with patient-derived biopsies. And indeed, there are also other applications using the same organ-on-chip platform, using human biopsies as is, or spheroids or organoids. So at the end, what we typically do is to grow and culture this three-dimensional cancer model that can be built co-culturing different cells, of course, the tumor cells, but also eventually in combination with these mesenchymal stem cells and fibroblast and endothelial cells. Endothelial cells are in some cases also used to create a sort of endothelial barrier where the tumor grow mimicking the vascular wall. And this is really important for studying also the infiltration, the tumor. So at the end, in the chamber of the organ on chip, you have an insert with a permeable porous membrane, which can be functionalized in these endothelial cells. And then on top, the three-dimensional tumor model. And as I will show you later, there is also the possibility to put in circulation immunocells together to the drug. So really, you can modulate and increase the complexity of the human biology in vitro by replicating exactly the crosstalk among different cells as what happened in animals, but using a fully humanized in vitro approach. In terms of the readout, in our tests, typically we monitor the viability and cytotoxicity of our cells, and this is really useful to have an organ-on-chip platform which is optically transparent and a plate which enable the real-time monitoring under the microscope. Moreover, we have the chance to sampling the media in the tumor niche, in the tumor compartment of the organ on chip, but also in the compartment mimicking the bloodstream. So the possibility of having two separate sampling of the media opens also the new opportunities in terms of measuring the gradients of cytokines, which are the mirroring of the inflammation. And of course, in the field of drug discovery, also the possibility to really measure the concentration of the drugs. At the end of the experiments, we have anyway the chance to pick up the tissue samples for further investigations, meaning histology or omics analysis. So if we can summarize the value proposition of this innovative approach is the possibility to work with patient-derived samples towards also an approach of personalized medicine, the possibility to co-culture tissue samples with immunocells, which are placed in a fluid dynamic condition properly resembling the capillary blood flow condition for more predictive results, using, of course, a 3R approach, finally reducing also time and cost for end users. In terms of drug efficacy, I consider also an important added value, the possibility to put the drug not immediately in the media containing the tumor model, but in a flow, in a circuit mimicking the blood flow, so mimicking the real systemic drug administration. This is extremely important because the kinetic of diffusion of the drug into the matrix, into the tissue, and therefore the readout of drug efficacy is different if you have such administration of the drug, which is more close to the in vivo condition. And therefore, what we decided to do, it was a comparison with the animal model, which still represent a gold standard for some assays like drug efficacy assays for pharma companies. And so we wanted to use the same drug, in this case it was a cisplatin anti-cancer drug, the same tumor model, and we used an ovarian tumor model. In terms of the dose of the drug, we used the plasmatic drug concentration observed in animals, and we used the same concentration in vitro by using the organ-on-chip MIVO that we have developed, and also a static condition, meaning the same tumor model cultured in a wild plate, so in a steady condition, which still represent another gold standard for scientists. What we did, it was the analysis of the proliferation state of cancer cells. In this case, we stain in green proliferating cells, and we stained in red the cells, the dead cells after the administration of the drug. Interestingly, after the application of the drug in the 3D tumor model cultured in steady condition, we observed still many proliferating cells, especially in the inner part of this millimetric tumor model, and some dead cells, especially in the external part of the tumor. Interestingly, the same tumor model cultured and treated in the organ-on-chip MIVO displayed after the treatment very few proliferating cells and many dead cells, especially in the homogeneously dispersed in the overall matrix. So the hypothesis behind this strong difference between static and dynamic condition was a difference in terms of diffusion of the drug. So our hypothesis is that in static condition, the drug is able to reach the matrix, the tumor matrix, kill the external cells, and then these dead cells generate, together with the matrix itself, a sort of barrier of the continuous diffusion of the drug into the tumor. On the other side, using an organ-on-chip, a fluid dynamic organ-on-chip, where the flow is driven by a pumping system setting up to mimic the bloodstream as in vivo, we have a continuous diffusion of the drug as what happened in vivo. And indeed, we found dead cells homogeneously dispersed in the matrix. And this is indeed what happened also in patients, because cisplatin is used in the patients. This conclusion was also successfully validated using computational fluid dynamic simulation. So we simulated the flow containing the drug, which diffused the drug into the tumor cells. And the take-home message here is that when you have tumor cells cultured in monolayer, so the red lines, static or dynamic condition, it doesn't matter in terms of diffusion of the drug. So it is also pretty intuitive. When you have cells in a monolayer and the drug placed in the media, the drug effectively reach the cells and induce a sort of cytotoxicity response. But when you go on and move on a three-dimensional tumor model, which is, by the way, extremely important to, again, keep the real heterogeneity of the tumor that we observed in patients, well, in that condition, in three-dimensional condition, which are the blue lines, it's really important to introduce also a flow for having the proper diffusion of the drug, but similarly, you can consider the diffusion of the nutrients or growth factors into the matrix. Otherwise, the diffusion is really limited by the matrix itself. This is the reason of why we obtain the same regression of the cell viability or the tumor mass in animals and in the MIVO. So animals are the black bars, and you see that we have a regression of the tumor up to 50% after 3 weeks in mice. And the same regression was observed also in the MIVO organ-on-chip already after 1 week. This is also another important, extremely important results, especially for CRO and pharma companies, which is the possibility to drastically reduce the timing of the drug efficacy assay. Interestingly, from a scientific perspective, you see that when you use a static condition, the drug efficacy assay is not predictive at all, because you observed in the gray bars a sort of chemoresistance, which is not in line with the clinical data. So this paper, these results are extremely important to demonstrate that it's possible to reduce the use of the animal models, at least for some assays like this one, because it's possible to obtain exactly the same results with a strong reduction of time and cost. By the use of the organ-on-chip and cancer models was also used for studying the spreading of the tumor itself. So we know that in almost all cases, the primary tumor can be also removed surgically, but the real challenge is to stop the progression of the tumor and the spreading of the tumor cells which leave the primary tumor intravasate, so pass across the endothelial barrier and become circulating tumor cells and then reach other tissue target. So in this paper, we demonstrated it is possible setting the fluid flow like the capillary blood flow to mimic the behavior of cancer cells, which at a certain time decided to leave their condition and becoming circulating tumor cells. Moreover, we also demonstrated that the viability of the circulating tumor cells is really affected by the shear stress induced by the flow, opening also new therapeutic and pharmacological perspective, meaning modulating the shear stress and the viscosity of the blood as a therapeutic approach in the field of cancer. And this experimental approach, open this possibility. So at the end, it's possible to put in circulation the tumor cells and observe their different behavior under different blood flow conditions, and also test the efficacy of new therapeutics in this specific condition. In the picture, you can also see the possibility to have multiple replicates of the same experiment in the same tray, so in a very compact way. Moreover, the organ-on-chip we have developed enabled to fluidically connect multiple chambers. This is extremely important in cancer because, as I mentioned, you have the possibility to close the circuit and complete the overall journey of tumor cells, which may leave the primary tumor, such as breast cancer, and become circulating tumor cells, and then reach a secondary chamber where we have a secondary target tissue like bone, which is one of the most common target of metastasis. So again, also in this case, it's useful, first of all, to study from a research perspective why and under which condition breast cancer cells and in general cancer cells are more aggressive in terms of infiltration of different targets. And from a pharmacological perspective, it's really important for testing the efficacy of new therapeutics. But the use of the multi-organ chambers has been successfully validated and used also for testing simultaneously the efficacy on target of an anti-cancer drug and the cytotoxicity of target, like the hepatotoxicity. So when we already know from the previous toxicity assays the target of toxicity of a certain drug, it's very useful to test different doses of the treatment, co-culturing in the same organ-on-chip the target of the therapy, but also the target of the off-target cytotoxicity, like in this case liver, but similarly you can figure out cardiac tissue. In this case, you have the chance, as we demonstrate in this paper, to really recapitulate in vitro the interplay among different organs, which at the end affect the behavior of the cytotoxicity, because there is a sort of cross-protection among different organs. But in my opinion, one of the most important opportunities by using this approach, meaning organ-on-chip with three-dimensional tumor models, is in the field of the immuno-oncology, because here we really need to have immunocells viable in circulation, exactly as what happened in vivo. So immunocells intrinsically are fluid dynamic circulating cells, and the idea is to properly resemble the condition affecting their behavior. In this case, we used a different tumor model. In particular, we used a pediatric, very aggressive tumor model, such as neuroblastoma. And we used two different cell lines also to validate the robustness of the approach. So we also, in this case, we used a matrix-based polymer where we seeded our cells. We successfully checked the viability of the cells. You see also the dimension, which is pretty big, of these tumor models. We checked the proliferation. Okay, so I listed some of the most important drugs under development in the market by some of the most important pharma companies, which are using exactly the PD-L1 immunocheckpoint path. And in particular, using our tumor model, we checked the proper upregulation of this target after the administration of the interferon gamma, which is typically released by immunocells. And nicely, we have seen the proper upregulation of PDL1, PDL2, and B7H3, and downregulation of PVR. So the take-home messages here is that these three-dimensional tumor model are also really reliable, and they present the proper immunocheckpoint expression as in vivo. This is extremely important for then co-culturing this tumor model with immunocells in circulation. And in this works, we used a subpopulation of immunocells, in particular the natural killer cells, but there are other partners involved in these studies that are using the overall PBMC from patients. So there are different approaches under development. And nicely, since, as I said at the beginning, we have the chance to sampling the media separately in the tumor compartment in the immunocells flow, we had the chance to sampling the media in the tumor compartment and quantify, if any, the amount of the immunocells able to leave the flow, win the gravity, and going up close to the tumor. And this percentage was in line with the clinical data, so was about 2% of the total amount of the immunocells placed here. And nicely, when we didn't use any tumor model, so the negative control, we found that a negligible number of immunocells were able to going up. And when we had the positive control that was the same extracellular matrix without tumor cells, an increasing of the serum, which is a strong chemoattractant, also in this case, the amount of the immunocells able to extravasate was significantly lower. So we demonstrate that the natural killer immunocells extravasation was really tumor specific, exactly as what happened in vivo. So extremely reliable and predictive results. Moreover, since we have the chance to collect the tumor easily from the organ on chip and carry out further investigation, we did some confocal imaging, and we found the immunocells here stained in green, very close to the tumor cells stained in red, and in particular, the apoptosis of tumor cells was significantly higher in this condition. So immunocells were able to survive under flow, extravasate, enter into the tumor matrix, migrate close to the apoptosis, as what happened in vivo. Moreover, from the immunological perspective, it's really important to have the possibility to sort the population of the immunocells infiltrating the tumor, and we found that this population was less CD16 positive compared to the other cells, and the CD16 positive is a marker of aggressiveness of the immunocells. So, and this similar approach is used also for co-culturing a patient tissue explants with immunocells from the same patient for selecting the more effective immunocells and selecting immunotherapies for the specific patient. In conclusion, I wanted to stress the fact that at least for this application, which is the oncology and immuno-oncology, the fluid flow is really mandatory, especially when you have a three-dimensional culture. And this is possible using this type of organ-on-chip platform. The combination of the immunocells on one side and three-dimensional tumor models on the other sides enable to foster the translational research in this field, and also the drug testing towards a personalized therapies approach. And the organ-on-organ connection enables to properly investigate the crosstalk among different organs, which is at the basis of many systemic diseases, and also for testing the on-target and off-target efficacy assays. I want to thank all the people involved in this study, and here you can find also my email for further questions and the grants supporting this research.
USDA is an equal opportunity provider, employer, and lender.